Introduction. Microbes or microorganisms are tiny living things that are invisible to our naked eye and can be only seen by using microscope. They can be found in everywhere, such as in water, soil, or air. Microbes can be grouped into 6 types, which are bacteria, protozoa, fungi, algae, archaea, and virus. Many bacteria live in our body, such as in gut, nose, mouth, and skin. Some microbes can cause disease when transmitted by bacteria, virus, or fungi. In ocean, marine algae produce most of the Earth's organic matter and at least half of the oxygen we breathe. For example, cyanobacteria, one of the oldest life forms that help create Earth's oxygen atmospheres. Extremophile are microbes that survive in extreme conditions, such as very high temperature, by adapting with unique genetic mechanism. For 5 kingdom classification, this was introduced by R.H. Whittaker, who is categorized life based on their characteristic. The first is Animalia. It is highly differentiated tissue and specialized bodily organs. It is complete with nervous system to coordinate body activity and responses. It does not have cell walls, but they do produce reproduction systems such as ovum and sperms, for example, birds and fishes. The second is fungi. It is eukaryotic, that the body made up from haploid and thalloid, while the cell walls are made up from chitin, for example, mushroom and yeast. The third is plantae. It is differentiated. The plant body can be differentiated into various sections such as roots, stems, and flower. It can be classified by observing the differentiate of plant body has vascular tissue, where the classification based on presence of seeds, thus the seeds are in cases within fruits, for example, mosses and ferns. The fourth is Monera. It is a type of prokaryotic that is made up from single cells without nucleus. The cell walls are made up from peptidoglycan, and they reproduce asexually, can be binary fission or budding, for example, archaea and eubacteria. The last is Protista. It is a type of unicellular eukaryotic. It has a hair-like cilia to move, for example, flagella, or pseudopodia or amoeba. It can gain nutrient by autotrophic or heterotrophic. For example, protozoa and algae. Binomial nomenclature, it can be divided into 2 sections, which are genus and species. For genus, it is Candida, while the species is Albicans.
Now, let's get into the characteristic of Candida albicans. Firstly, its morphological characteristics. Candida albicans is a eukaryotic unicellular organism and is also a gram-positive, which means that it has thicker peptidoglycan layer compared to the gram-negative organism. Candida albicans can be differentiated from other members of this genus by the presence of chlamydospores and pseudomycelia. Chlamydospores are large formation at the end of the hyphae. It is round in shape with a thick wall and diameter of 7 to 13 micrometer. Additionally, Candida albicans can grow as yeast, pseudohyphae, or true hyphae. The difference between hyphae and pseudohyphae is that hyphae are thread-like structures found in filamentous fungi, while pseudohyphae are chains of newly divided cells of unicellular fungi. Next is the nutritional characteristic of Candida albicans. Candida albicans is a heterotrophic organism that obtains organic compounds such as sugar, amino acid, and lipids from its environment or host. It is also chemoorganotrophic, which means it utilizes carbohydrate as carbon and energy source. It also can metabolize alternative carbon sources when glucose is scarce, such as lactate, acetate, and fatty acids via gluconeogenesis the glyoxylate cycle and beta-oxidation pathways. Furthermore, Candida albicans is osmotrophic. It absorbs nutrients through osmosis. These characteristics indicates that Candida albicans is from the kingdom fungi.
Habitat of Candida albicans. Candida albicans normally live as part of the normal flora in the human body, especially in the oral cavity, gastrointestinal tract, vaginal area, and moist skin surfaces. This image shows candidiasis, a fungal infection that is most commonly caused by Candida albicans. Under certain conditions, such as a weakened immune system or antibiotic use, Candida albicans can overgrow and cause infection. For reproduction, Candida albicans reproduce mainly through asexual reproduction. It usually exists in the yeast form, where reproduction occurs by budding. In this process, a small bud forms on the parent yeast cell, grows, and eventually separate to form a new cell. Under certain environmental conditions, Candida albicans can change its morphology into pseudohyphae and true hyphae. This filamentous form help the microorganism spread and invade host tissues, increasing its pathogenicity. In addition to asexual reproduction, Candida albicans is also capable of sexual reproduction, but this occurs rarely and is not considered significant in clinical infection.
Now, I will be talking about the significance of Candida albicans. Candida albicans actually can cause infection when the immune system is weakened or when the normal microbial balance in the body is disturbed. Under normal condition, it exists harmlessly as part of the normal human microbiota. However, when these controlling factors are disrupted, Candida albicans can multiply excessively and cause disease. So because of this behavior, Candida albicans is actually classified as an opportunistic pathogen, which means it does not usually cause infection in healthy individuals, but becomes pathogenic when favorable conditions are present. So the significance of Candida albicans lies in its ability to cause infection due to several risk factors. One of the major factors is a weakened immune system, such as in patients with HIV or AIDS or cancer patients that undergoing chemotherapy where the body is unable to effectively control fungal growth. The next factor that we have is the prolonged use of antibiotics. Antibiotics will reduce the population of normal bacteria that compete with Candida albicans, allowing the fungus to overgrow. The next one we have is diabetes mellitus. Diabetes mellitus also increase the susceptibility to infection because high blood glucose levels promote fungal growth and impair immune response. In addition, use of immunosuppressive drugs such as corticosteroids or post-transplant medication weakens the immune system and increase the risk of candidiasis. Furthermore, medical devices such as catheters and implants provide surface for Candida albicans to attach and form biofilm, making infection more persistent. Next, we'll be looking at the features that increase the pathogenicity of Candida albicans. One of the most important features is dimorphism. Dimorphism is actually the ability to switch between yeast and hyperforms. This ability helps fungus invade host tissues more effectively. Next, Candida albicans can also form biofilm, which protect the fungus cell from antifungal drugs and the host immune system. In addition, it has adhesion mechanisms that help it attach to host tissue, allowing colonization and persistence. The last pathogenic features is enzyme production, including protease and phospholipids. These enzymes damage host cell and tissue, facilitating infection and spread. These pathogenic features of Candida albicans can cause several common diseases, such as oral candidiasis, also known as oral thrush, is characterized by white patches in the mouth and throat. The next one we have is vaginal candidiasis, or yeast infection that cause itching, irritation, and thick white discharge. Candida albicans can also cause cutaneous candidiasis, which affects the skin, particularly in warm and moist areas such as the groin and armpit. In severe cases, it can cause systemic candidiasis, also known as candidemia, where the fungus enters the bloodstream and spread to vital organs, making it a life-threatening condition.
Next, I will talk about the example of Candida albicans in commercialized product. There are three examples, which are Candida albicans are used for skin test antigen called Candida. Secondly, it is used for quality control testing in making a product. Lastly, it is used for biosurfactant in industrial microbiology as detergent and cosmetic for our daily life. Next, I will talk about the process of Candida albicans in commercialized product. For Candida albicans skin test antigen, product name Candida evaluates delayed type hypersensitivity and assess the immune response to Candida albicans with extract of Candida albicans as the active ingredients. For quality control testing, the standardized strands of Candida albicans are used for verification of culture media performance and validation of diagnostic assays. This helps to control the product in manufacturing it. Lastly, for biosurfactant product of Candida albicans, it is used in industrial microbiology. Biosurfactants such as sulfurolipids are biodegradable and environmentally friendly, and although Candida albicans has been studied, it is not typically used for industrial production.